and welcome to today's Public Health Ontario's microbiology presentation on a very cool and a very important topic, CRISPR diagnostics for tackling antimicrobial resistance. My name is Vanessa Tran, and I'm a clinical microbiologist at Public Health Ontario, and I have the absolute pleasure of moderating today's session. Before we begin, there's just a couple of housekeeping items. So please don't use the chat pod because it's been disabled to limit any distractions during the presentation. Please use the Q&A pod if you have any questions during the session. A discussion and question period will follow the presentation, but if at any point during this session you experience any technical issues, please email capacitybuilding@oahpp.ca. I would like to state that as the moderator of the session, I do have some conflicts of interest to disclose. I am the board of the Canadian College of Microbiologists and Canadian Association for Clinical Microbiology and Infectious Diseases. I'm also a co-investigator or collaborator on grants funded by CIGR, PHAC, and Gilead. It is now my pleasure to introduce the speaker for today's presentation, Dr. Nicole Weckman. Dr. Weckman is the Paul Cadario Chair in Global Engineering at the University of Toronto. Her research focuses on developing novel point-of-care technologies for diagnosing diseases and monitoring outbreaks of drug-resistant infection. Her research group develops sensitive and quantitative sensors at the interface of cell-free synthetic biology and microscale and nanoscale sensing systems by focusing on engineering design for clinical and commercial translation. Her interests are in developing low-cost and sustainable diagnostics that can help improve health equity. Thank you so much, Nikki, for presenting at Micro Rounds today, and I really am looking forward to your talk.
Thank you so much, Vanessa. So as Vanessa mentioned, I'm Nikki Weckman, and I'm very excited to be speaking here today and getting to know all of you. I just started my research group here at the University of Toronto in January, and I'm cross-appointed between the Center for Global Engineering and the Department of Chemical Engineering there. And as mentioned, my research is really focused on developing new sensing system to help us tackle some of these big global challenges that we're facing, like access to healthcare and environmental conservation. So I'll start with a few disclosures of my own. I'm a co-founder and shareholder and the Chief Technology Officer at 52 North Health, which is a medical diagnostic company. I will not be presenting any work related to the company today. And a disclaimer that I created this presentation, so this is my own research talk. And I would like to start by thanking the many people who have funded, mentored, and partnered with me so far in my career. I really believe that science and engineering are team sports. You can see in the top left hand corner there, this is my young group at the University of Toronto. And we're currently working on projects that are funded by EPIC, the Center for Global Engineering, and NSERC. But I'll also be talking to you about some work today that I did during my postdoctoral studies. And so I did a postdoc at the Beast Institute in the Collins Lab, and this is where we started the work on detecting multidrug resistant Candida auris. So I'll present some of the unpublished first results from that study today, and we can talk about it a little bit. And I'll also talk to you about some work that I did at the Kaiser Lab at the University of Cambridge, where I worked on a project in nanopore sensing, and that was funded by Oxford Nanopore Technologies. One of the areas I'm particularly interested in at the moment in terms of global challenges is the increasing challenges associated with antimicrobial resistance. And this is an area that's already having a profound impact in healthcare, but is also a challenge across many different fields, so sustainable agriculture and environmental protection as well. And you can see that it's projected to have tremendous impacts on our society in the coming years and decades, both in terms of health and well-being, as well as in terms of economic costs. Thankfully, there are a lot of bright minds working on this, and global experts have agreed on some key strategies for helping to tackle antimicrobial resistance. In particular, diagnostics have the potential to play a key role in a few different areas. But they'll have a particular impact, in my opinion, in improving antibiotic stewardship. And this is because antibiotics are often given based on symptoms because it can get days to get culture results back. And so by the time these results are available, often the decision has already been made about which antibiotic to put the patient on. And that could be one that will be effective at treating that infection or one that potentially might not be. And so diagnostics are really critical to get before prescribing those antibiotics. So if you can get a really rapid result, you can then ensure that the right patient is matched with the right treatment. In Canada, we often have access to susceptibility testing and surveillance programs, but antimicrobial resistance is one of these big global problems that's really difficult to contain geographically. And around 47% of the population has little to no access to diagnostics. This gap is highest for primary care in lower and middle income countries, where as low as 19% of the population has access to diagnostics, especially affecting poor, rural and marginalized communities. I think it's important to acknowledge that lack of access to healthcare is not just a global phenomenon. There are many communities within Canada as well that struggle to get access to healthcare that they need and that's delivered in a safe and appropriate way to them. And I think as somebody who is designing technologies for healthcare systems, it's critical that we consider how these technologies will affect equity seeking and vulnerable members of our community, and try to make space within that design, development, and implementation for some of the lasting impacts of our actions in the past and some of the traumas that in many cases still exist. And so with that, I would like to acknowledge this land on which the University of Toronto operates, where I'm lucky enough to be able to set up my group to look into some of these issues. And for thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island. And we are grateful to have the opportunity to work on this land and to be able to conduct our research, which I hope will feed back into making equitable systems and protecting the wonderful earth that we live on and that we live with. So when we're talking about different types of technologies and diagnostics that can be used in global healthcare, it can be quite a range of different things that we're referring to. So on the right-hand side, you can see large, expensive, high throughput analyzers, complicated workflows. And these are often used in centralized labs like our hospitals, especially in Toronto. On the far left, you see a low-cost single-use technology. So for example, a home-based COVID test that we've all become increasingly familiar with. And in the middle, you can see an example of the Oxford nanopore sequencing device. And this is an example of an emerging technology that is rapidly being adopted and used in a broad range of settings. And it's really interesting because you only need laptop power, so you can scale these and have many of these in a big lab, but ultimately they can be very portable and used for analysis out in the field. And I think that this class of technology is going to have a growing impact in the diagnostics field in the coming decade. My research really focuses on inventing new diagnostics and new techniques that we can use in these last two categories that can provide the same or improved quality of results as in our central labs, ideally, but which I believe have the potential to help decentralize and democratize healthcare. And as I said, sometimes we have this tendency to assume that the field of diagnostics is solved, that there are great technologies out there that exist, and this is true. But I think we've also all lived through some of the challenges in diagnostic development in the past couple of years. There are a set of commonly accepted criteria for a fit-for-purpose diagnostic, the reassured criteria. And these include things like sensitivity and specificity, which is that core focus, and we tend to really focus on that in engineering design. But also include things like usability and affordability and connectivity. And it remains a challenge to meet all of these criteria, and particularly to meet all of them without making sacrifices. So often they're interrelated, and you have to make trade-offs in design. I think that the next generation of diagnostics should also aim for two additional targets of being multiplexed and quantitative. And in infectious disease, multiplexing could be detecting multiple diseases or pathogens, multiple resistance genes, for example, or even incorporating different types of biomarkers. So I'm really interested in looking at both nucleic acid and protein biomarkers from the same sample so that we can get richer data sets. And quantitative or semi-quantitative results, sometimes these are necessary, but I think there are certain cases where it would be really helpful to be able to tell, for example, viral load. Fortunately, unlike for COVID, there are no diagnostics for many infectious diseases, and especially for tracking antibiotic resistance. And this is especially true in terms of low-cost portable formats that can be used around the world. So I'll tell you a little bit about some of the work that I'm doing to try to address some of these issues, and hopefully you can see that we're making progress towards being able to integrate and troubleshoot a lot of those reassured criteria as we're developing new diagnostic formats. My lab is working at the intersection of three main areas. I use synthetic biology to create sample-to-answer assays for detecting COVID, and for multidrug resistant pathogens like Candida auris and gonorrhea. I also research micro and nanoscale sensor development and how we can expand nanopore sensing applications beyond the sequencing that some of you may be familiar with. I won't touch too much on MEMS development today, but there's a whole element of my research that looks at these silicon micro devices that I won't have time to go into. And finally, I study how we can best approach designing diagnostic sensors for successful commercial and clinical translation. And we can publish as many papers as we want on new sensors, but to me, it doesn't really help if we can't get them into a format such that the people who need them can then access them. And so I'll touch at the end a little bit on some of the areas that I think it's important to consider in terms of design early on in the progress. But I will start by telling you about CRISPR-based diagnostics and how I've been using them specifically to detect multidrug resistant Candida. I want to start by highlighting two key advances in synthetic biology diagnostics in the last decade so that we can kind of all start on the same page. Synthetic biology is a discipline that aims to re-engineer the processes that occur very efficiently in nature. So, for example, our ability to make copies of DNA, to transcribe DNA into RNA and back again, and also to translate RNA into proteins. And one of the big advances in using synthetic biology for diagnostics in limited resource settings in particular was the demonstration that you can remove this transcription and translation from a cellular environment and freeze dry it into a pellet or a piece of paper. And this is a paper that summarizes some of the advances in that area. Remarkably, if you freeze dry this transcription translation machinery into a piece of paper, upon rehydration, so when you add a sample, for example, it will still work as you expect it to. And Keith Pardee published some of the seminal work in this area here back in 2016, 2017. In cell freeze-dried synbiodiagnostics, demonstrating the use of toehold switches in particular, which is this RNA switch up here for detecting things like Zika and dengue. And then in 2017, 2018, a series of papers were published that demonstrated that we can also use CRISPR-Cas systems that are of gene editing fame for diagnostics as well. And Cas enzymes are proteins, and they very specifically recognize a nucleic acid sequence through the design of a guide RNA. So on your screen here, you can see there's this pink region of a guide RNA right here that is complexed into the Cas, and then a purple structural element. And you can design that guide RNA pink sequence to be complementary to the target that you're trying to detect, whether that's RNA or double-stranded DNA. Now, Cas12 and Cas13 have this really cool property in that they can be repurposed into CRISPR-based diagnostics by taking advantage of a conformation change that happens when they recognize their target sequence. So when the Cas13 or the Cas12 recognizes the target sequence, there's a conformation change that happens that activates a collateral cleavage mechanism. And this allows us to non-specifically cut other nucleic acids nearby. So for example, this reporter molecule. And this idea, this concept was published in a couple of papers. The detector paper really focused on the use of Cas12, and that came out of Jennifer Doudna's group. and Sherlock, which came out of the Boston area, Jim Collins and Feng Zhang, looked at Cas13 and in the first instance was targeting RNA. But these types of assays are a little bit more interchangeable now. The lines have been smeared over the years. So I want to tell you a little bit more about the specific assay that I've developed. So we take advantage of the Cas12 binding specificity. And what we've done is we've designed a guide RNA that can target the specific 20 base pair sequence. So for example, if you look at this Cas molecule right here, you have a guide RNA that is binding to a double-stranded piece of DNA. And when it binds, this Cas molecule undergoes that conformational change, and it cuts a reporter that has a fluorophore molecule and a quencher on it. So when this is attached here, the quencher it turns off the fluorescence essentially. And then when it gets cut, because you separate the quencher from the fluorophore, that fluorescence signal will then turn on. By changing the two functionalities of this reporter molecule slightly, you can also put this into a lateral flow format such that when the reporter is cut, you get 2 lines on a lateral flow strip, whereas if it's uncut, you'll only get a single line. Now, we couple in an isothermal amplification step to this. In our case, what we're using is something called RPA. You can use RT-RPA if you're wanting to deal with RNA. This isothermal amplification is great because you don't need to do thermal cycling, for example, what you would expect out of PCR. And so you can take this out of a setting. It happens at 37 degrees. You don't need fancy equipment to be able to do that. And it improves the sensitivity quite significantly, which I'll show you in the next slide. However, these isothermal amplification steps often tend to limit the quantification. So they're exponential amplification steps. And so you lose the ability to tell how much was in the sample, but you get a very sensitive yes/no answer to the presence or absence of a target. So I wanted to tell you a little bit of an overview about where we're going here. So where are the future of these projects taking us? So when I started here at the University of Toronto, I started up a new collaboration with Dr. Rob Kozak up at Sunnybrook and some of his other collaborators as well from across Toronto. And we're expanding the ability to detect Candida species. So we're developing more of a lab-based assay at the moment that will be able to detect the 5 key Candida pathogens, as well as building out our ability to detect different resistance mutations, with the aim of then doing some clinical lab testing on a variety of samples that are kind of coming through hospitals in the Toronto region. And we have a great set of collaborators there with Rob and many others from across Toronto. But if you're interested in this project or the next project I'll talk about, please do reach out. The other project that we've just started, and I'm not showing you results on this today because we're in the very early stages of development here, is detecting multidrug resistant gonorrhea. And this is an area that is really important, particularly in the global health context. The WHO and FIND have released 2 target product profiles back in 2020 to improve our ability to detect gonorrhea, and particularly to build in antimicrobial resistance detection for gonorrhea. And so we're working with some great partners on this project, where we'll design the initial designs of a CRISPR-based diagnostic for antimicrobial resistance and gonorrhea. And then engage with people living in refugee camps. So we have a partner in Uganda and healthcare practitioners who try to help people in those settings. And we're collaborating with Professor Carmen Logie, who is the CRC in Global Health Equity and Social Justice with Marginalized Populations. And I'm really excited about this partnership because Carmen is fantastic and really looks at health stigmas, especially as relates to STI testing and access to STI testing in global health. So I think we have the potential here to develop something that will really address some of those unmet needs and hopefully break down some of the barriers to testing. So I wanted to give you a few examples of the final packaging because the Candida project is in fairly early stages at the moment, but we have published a few papers in areas and detecting other infectious diseases. And so I wanted to show you how you can implement these in really easy to use formats. So one example of something that we published a couple of years ago was our SARS test that detected both SARS-CoV-2 as well as identified which variant was causing the infection. And we did some clinical sample testing on that. Please see the paper if you're interested further in the performance-related metrics on that. But in terms of the packaging, I thought this was a really cool way to wrap up the diagnostic technologies. So it used a saliva sample. So you put 2 mils of saliva into this chamber on the left-hand side there. Essentially that ran the sample through a filter while heating it to deactivate some of the other things in the background of the saliva that might interfere with the assay. And then the user was required, after putting the saliva in, to do only one other step, and that was to transfer that filter over to these lyophilized pellets on the opposite side of the diagnostic there. This all happened, so there was a heating step to deactivate some of the things in saliva, and then a small heater here that kept the reaction at about 37 degrees, but we also tested this at room temperature, and these enzymes tend to work well at room temperature as well. You can see the final implementation of this. So this is all available online. We included all the CAD models, so you can 3D print this for yourselves. It costs about $15 to make this whole device. And once you have the electronics, so it's battery powered, it has 2 small heaters and some LEDs, and basically you can see the fluorescence readout here. These pots are modular, so you can swap it out. So you can keep the electronics, for example, and the rest of the device, and swap out what you're trying to detect. And everything is recorded in a smartphone app so that we don't lose any of that data collection. We wanted to see how many different variants we could detect, for example, and we were pushed to kind of expand out the platform a little bit. And this is both a win, but also one of the things that I'm trying to address, and I'll talk a little bit more about with the nanopore system. But essentially, the way of multiplexing in this instance is simply to add more pots. So you split your sample up into further pots, and you can see that you can test for 3 different variants and a negative control there to make sure that we're not getting false positives. You can multiplex this system, but only to a certain extent. So you can imagine you're going to hit a limit where it's just not feasible to keep adding pots and to keep track of what they are. I wanted to show you some examples outside of our lab as well to give you more of a balance of some of the cool things going on in the field. So this was a plate reader. So if you want to more highly multiplex, for example, this was something that came out of Keith Party's lab, where he actually took this out into the field. And it is a toe-hold switch based sensor that detects Zika. There's a color change that happens from yellow to purple based on identification of the target sequence. And they've built this Plum reader, which is portable, low cost, user friendly, and multimodal. And so you can see that this reader here is not your typical plate reader. It's something that you can easily take out into the field and carry around. So it's all powered based off of a Raspberry Pi computer. A more lab-based method is the Carmen system. And this is a highly multiplexed assay that simultaneously detects 169 human-associated viruses. And one of the key advantages of this is not just the ability to multiplex, so it's a microfluidic chip-based system where you mix different droplets. So you can, this is very highly multiplexed, but it also reduces the reagents needed for each test. And so the reagents per test are decreased by more than 300-fold by using these small volume reactions. And so you can test very high throughput test a lot of different things in very high throughput in this simple chip. And then one last interesting example that is looking at making these methods more quantitative is the digital CRISPR assays. And basically what this involves is separating out, rather than having a large pot full of all of your different reagents, you separate out the entire reaction into tiny picoliter size wells. And each one of these wells is either going to have a target molecule or it won't. And so if it has a target molecule, you can see the fluorescence turns on. If it doesn't have a target molecule, it will stay dark. And so based on the proportion of these wells that light up, you can then get quantitative information about the levels of your targets. And the sensitivity is maintained here because you're still amplifying, you're just doing it on a single target in one of those wells. So I want to talk to you about the nanopore system just very briefly in the last couple minutes, so that I can show you some of the efforts that we're making in this area, because I think it has the potential to expand both the ability to multiplex and to make these things quantitative. And in its very simplest, a nanopore is just a nanoscale opening, and you can use different types of nanopores depending on what you're trying to measure. But you want your nanopore to be just larger than whatever molecule you're trying to measure. So in the case of sequencing, essentially you have a nanopore here that is 1 to 2 nanometers, and you're pulling through a single strand of DNA. So you can get that really sensitive sequence-based information. But in my case, what I'm using is a slightly larger glass capillary nanopore that is about 20 nanometers in diameter. And that allows you to pull things through, for example, proteins complexed with a piece of DNA. And this allows you to collect very sensitive size, shape, and binding interactions in a very simple system. And so I'm going to show you a quick video here of exactly what I was trying to do. So in this case, we were using Cas9 again. But we were trying to label different pieces of DNA with barcodes made out of these dCas9 molecules. So for example, if you have a mixture with different types of DNA in the background, so you have your blue and your green DNA, you can design dCas9 molecules such that you have, for example, 3 Cas9s that will bind to the blue target and 2 that will bind to the green target. Now, because these are dead forms of the molecule, they just bind, they don't cut anything. Now, once we've labeled these sequences, and those are going to be very sequence specific to whatever the green DNA is or whatever the blue DNA is. Now, the way the nanopore works is you apply a voltage across the pore, and this causes an ionic current to start flowing. So the salt in the background buffer is going to start going through the pore. When something like a piece of DNA goes through, it blocks some of that salt flowing through the pore. And that blockage that's going to happen is very characteristic of the DNA that's going through. So you can see when you have these Cas9 molecules on it, you start getting peaks in those DNA traces. And so, for example, the one that's coming up, you're going to see 3 peaks in your current trace that correspond to where those labels of dCas9 are. Now, because you can control where you're placing these labels and how many you target to each different type of DNA, you can create these really unique barcodes and then collect a lot of events so that you can get statistically relevant information. And the motivation behind this project, it was sponsored by Oxford Nanopore Technologies to try and get more rapid sample to result answers when in cases when you didn't need whole genome sequencing. So if you just rapidly want a yes or no answer, is this DNA present in my sample? Creating these really unique ionic current trace sequence patterns is going to be a much more rapid way to be able to tell that than actually sequencing everything in your sample. And so we basically started from the very beginning on this project and showed that we can, in fact, target these dead Cas9 to certain locations on our DNA. And so this is an AFM image on the left that shows those little blobs. Those are the Cas molecule attached to these really long pieces of DNA. And we're just using lambda DNA, so a dummy DNA here. We then did exactly what I showed in the video, where we designed a few Cas9 molecules to target different sequences in very characteristic patterns. So in one case, we were using lambda DNA, and we targeted 2 Cas9s to those sequences. And so we got these double peaks. And the other case, we were using T7 DNA, and we targeted 3 Cas9 to that one and got these triple peaks. Now, what we found was that despite mixing all of these Cas9 molecules in there, you can see that only a small percentage of events, so about 25 to 30% of events, were actually correctly labeled, right? So it didn't have 100% efficiency on picking up these types of DNA. And one of the challenges that we found when we tried to multiplex this, so mixing the lambda and the T7 into the same sample, was that even if we mixed them in equimolar concentrations, we often had a much higher percentage of events labeled with doubles versus triple barcodes. And I think there's a couple of things going on here. So number one is the efficiency in having all 3 Cas molecules bound to the same piece of DNA at the same time is going to be lower. And the second thing is that we had crosstalk between these barcodes. So essentially, if you miss one Cas from the triple barcode, it can be identified as a double barcode. So we did a lot of work into how we can design and test better barcodes, but also how we can make sure that we're selecting good Cas9 that binds very strongly to the DNA. And to do this work, we created synthetic DNA constructs that allowed us to very carefully target. In the interest of time, I'm going to skip a couple slides here, but that allowed us to build in multiple binding sites on a single DNA structure. So you have a backbone of DNA here, and then you have these binding sites in yellow. So in this case, they're all the same, whereas in this middle case over here, you can see that we have one binding site that's red, one that's green, and one that's blue. And we can then screen, for example, if you have this target purple molecule that is trying to bind to a specific sequence, how susceptible is it to changes in that sequence? So if you place mutations on different sites, or if you have off-target sites that you're worried about, you can change one of these sites to test its specificity to your target sequence versus your off-target. So in our case, what we did was we used this to study how susceptible it was to mutations at different positions related to the PAM site, because we were also interested in this mutation detection. And we found results that were really interesting in terms of where exactly, how far out you had to get from your PAM site before a mutation would not be able to be detected. So you can see the background here, the cross-reactivity between that mutation and other mutations starts to raise. Whereas if you have a mutation that's very close to your PAM site over here, you're only detecting the one thing that you really want to detect. So we're working on building out this system a little bit more so that we can understand, and this will feed both back into our understanding of how Cas molecules interact with mutations, as well as our ability to design better guide RNA. At the very end, I'd like to spend the last couple of minutes here just talking about the translation of these sensing systems. And so I think there are a few areas that we're really interested in the lab. And this is, I love to co-create with either patients or clinical end users. So whoever is really going to be using these technologies at the end, it's important to get that feedback very early on. We also need to build in and start thinking about how digital platforms can be used for improved surveillance and for collecting a lot of this information. And so we have a strong focus on making sure that we don't lose information from the point of care sensors after the result has been delivered. I think from COVID, we realized that there's a lot of single-use plastics that are starting to be thrown in the bin. So can we start looking at how we can manufacture these more responsibly and in a way that we're not increasing the negative impact to our planet? And then also looking at reducing some of those policy hurdles. And I think that my point here is just that diagnostic devices never exist on their own. And so it's important to take a step back and to learn from experts in some of these pathways about how this device will be used and what the impacts of a positive or negative result are, so that we can take that into account when we're designing diagnostics from the very beginning, and we're not making problems with usability errors that will then trickle throughout the system and make everybody's life more complicated. I think it's a really exciting time to be working in this field because of some of the trends that have been happening through COVID in terms of decentralizing healthcare, and increasing the diverse diagnostic use cases. It's not just a case that you're using these in hospitals anymore, and starting to look at how we can continue investment in the field, because investment has trailed off a little bit. Diagnostic sensors have the ability to create an impact across a broad range of areas. So not just healthcare. I've talked to you about a lot of healthcare impacts, but actually we're looking at how we can use this type of sensing technology in things like the environment, agriculture, and food safety, and biomanufacturing as well, with the aim of really making a tangible impact to some of these huge global challenges that we're facing as a society. So I'd like to thank you for your time and engagement today. And I hope that I've inspired some excitement about new diagnostic technologies and ideas about how we can use these to improve equity and access to healthcare, and to tackling other SDGs as well. So I will end as well with an invitation. Please reach out if you have ideas or want to continue this discussion after today.
Thank you so much, Nikki, for that very informative presentation. We'll now move on to the Q&A segment of the event to address some questions from our audience. If you've not had an opportunity to do so, please continue to enter your questions into the Q&A pod, and we will address them as we go along. So we do have a couple of questions to start off with, and the first one has to do with performance and cost. So before getting to the question, which is, do you think combining CRISPR-Cas and carbon quantum dot could provide less costly and better signal-noise ratio detection? Can you provide just a super quick overview of what it costs? Like, what does one of your CRISPR diagnostic actually cost?
Yeah, so we did a full cost up that you can see in the My Sherlock paper. In the supplementary, there's a table of exactly the breakdown of every single cost that's in there. The total device there with that 3D printed setup and the heaters, for example, was $15. I think the cost per test was reduced to $6 for one of those little pellets, if you reused all of the heaters and stuff like that. Now, obviously, we have not, that was based off of purchasing research-based enzymes at non-commercial levels, right? So if you're actually manufacturing these tests, you can negotiate those costs to come down quite considerably, because you'll be buying enzymes in much larger batches, essentially, than we are for research purposes as well. But that is, I think, a useful starting point. And the other thing is, if you embed them in paper, some of the difference, right? So the difference between the $6 and the $15 can also be reduced. Could you repeat the question for me?
Sorry, it was whether or not combining CRISPR-Cas and carbon quantum dot could decrease the cost and increase the signal-to-noise ratio detection.
Yeah, so I think people have done some research, and there's some other people at Toronto, I think, that are working on this. And I wish I had reviewed that paper before this talk. So I think there are some performance advantages, and I think they can also help with multiplexing, because quantum dots, you have some additional kind of color responses that you can build in beyond kind of the traditional fluorescence detection. So you're able to detect more things at once. In terms of cost of quantum dots, I am not an expert, I have to say. So I don't know if I can address that, but I think there are definitely some interesting performance benefits to using a quantum dot output.
Okay, so I'm just going through the questions. So one attendee just wants to thank you for your use of single-use plastics, which I will echo. I think acknowledging the environment's impact of a lot of our laboratory testing often is is not done, and it is quite a large number of costing. So we have a couple of questions about translation. I also have a question about translation. So the question is about, what do you think the time would be needed to translate the work into a diagnostic, and what regulatory hurdles do you think there would be to bring these into the field?
These are great questions. So time to translate and regulatory hurdles. Right. So I will say I don't have any relation to these companies, but based off of the 2017-2018 papers, there were 2 companies founded that are working on translating these diagnostics. Sherlock Biosciences received an EUA for a COVID lab-based test, I think, during COVID. And that was I think the first diagnostic approval by the FDA, not approval, sorry, the first diagnostic authorization by the FDA for use of CRISPR-based diagnostics in labs. So that was a really big step and big moment in the field, I think. Now, an EUA is not a full authorization, and I think that the regulatory hurdles that we're seeing post-COVID have kind of come back into place, right? Because a lot of those tests that were kind of approved temporarily for emergency use during COVID are now trying to go through kind of the more rigorous process of getting them through the FDA. And that can take years. I think in Canada, we have additional hurdles as well. So from submission, so essentially in terms of translating these devices, you do all of the development work, you freeze the design, and then you start going into manufacture. And in order to be able to manufacture those, you have to show equivalence of 3 batches, take it through a clinical trial and a usability trial, and do a lot of analytical verification validation as well. And so that whole process can take at least 1 year, maybe 2 years, depending on how difficult it is to get these consistent batches in manufacturing, right? At that point, you have a regulatory file you can put into the FDA. FDA at the moment is taking at least a year to review a lot of these diagnostic technologies. And in Europe, it's even worse. IVDR is really backed up. And so you're looking at more than a year. And this makes it really difficult for diagnostics companies as well, right? Because it's expensive to go through this whole process. And then you end up just kind of sitting on your thumbs a little bit at the end of that, waiting for the regulators to get back to you. In Canada, I think that it's difficult because healthcare is then also a provincial system, right? I'm preaching to the choir here. And so there's actually additional kind of regulatory hurdles that you have to do or meet in order to get your technology into each province, right? And so I think it's something that we need to look at in the Canadian system is there a way that we can streamline some of those things such that it's not just a national regulatory hurdle followed by each of these individual provincial hurdles, but so that we can get some of these novel technologies out more quickly as well. Now, I will just say there's a bypass to this whole system in terms of translation, and that's if you can show that it's really reliable, you can implement it under certain smaller regulatory kind of barriers in individual labs, right? And so as long as you're not commercially selling it to people, you can implement these diagnostics less officially, I guess.
Thank you. So it sounds like there's a number of technical as well as administrative hurdles or challenges in order to commercialize or translate the, I guess, in research lab to diagnostics.
Yeah.
Just to follow up on that, the study that you did in New York with the Candida auris, was that done as a research project, or was that actually applied clinically? Like, did the hospital use your results to do their IPAC measures?
Yeah, so at the moment, that is just a research study, but I think they're probably very interested in continuing to use that as is Rob up at Sunnybrook, I think. And so I think longer term. Now, we still have a few things that we need to work out there. So one of the big problems, I think, and big challenges that CRISPR-based diagnostics are facing is, well, there are 2 of them. One is reproducibility, so making sure that every single enzyme reaction works exactly the same, which obviously is pretty critical if you're going to be implementing this for your surveillance screens. And then I've now forgotten what the second one is. Contamination. So you have to have, so once you've amplified these up, it is fairly easy to actually, you have a very high concentration of amplicons, so it's fairly easy to contaminate your space. So you have to be a bit careful about exactly how you implement it such that you're not getting kind of crosstalk or contamination issues in your lab. Now, that being said, I think that there's a lot of interest in implementing these. And so these are active areas that we're trying to sort out such that they can be used in surveillance efforts.
I'm going to ask a question because I think it's related. I'm going to pop to a question about the technology from the Q&A pod. So my question is, I think you've touched a lot on the translation implementation, and you showed the reassured criteria. I noticed in part of that criteria, it doesn't specifically address scalability. Can you just touch quickly in terms of the technology and how scalable it is?
Yeah, that's an interesting question. So if this was 5 years ago, I think probably I would say that there are going to be massive challenges, right? But I think actually we've seen that the biotech sector and the life sciences tech sector are moving very, very quickly. And so I honestly think that scalability is not a huge challenge with CRISPR at the moment. I think that making sure that we have good manufacturing practices such that it's very reproducible is going to be a really critical step. And then engineering out any ability to have cross-contamination between different things. But actually, you can see that a lot of these enzymatic reactions are actually commercially available already. So for example, RPA, the amplification, right? That's very scalable. It's used around the world. They can mass manufacture that. And I think in terms of ability to deploy, because I think that's part of the reassured criteria as well, the advantage that the CRISPR-based diagnostics have over a lot of maybe traditional immunoassay-based lateral flows, for example, is that when you freeze dry them into paper, for example, into a lateral flow format, they're very, very stable for long times. And so in terms of being able to scale deployment, I think that is a huge advantage that CRISPR has over some other technologies. They're completely cold chain independent, so you don't need to keep them in a fridge. They have really long shelf life.
That's a really great point. Okay, now, and I'm sorry to the individual that I jumped, but so the question is more about the technology. I'm wondering if any detection application for class 1 Cas proteins could be used for universal mutation detection across several species for low profile early screening. So I guess it's more of like an agnostic detection method.
Sorry, could you repeat that, Vanessa?
Yeah, maybe I'll just repeat it verbatim. Do you think there might be any detection application for class 1 Cas proteins, maybe a universal mutation detection across several species for low profile or early screening?
Oof. Off the top of my head, I'm not 100% sure, to be honest.
I think what the question is getting at is, are there other technologies that can be leveraged? Because I know you talked about multiplexing, but again, it's very targeted and it's limited to, in your system, a finite number that can be multiplexed. But is there a technology that could be used that could be a bit more universal in the detection of different types of mutations?
So there are other synthetic biology gene circuits that you can use in this area. I'm not entirely sure. I'd have to dig into the different Cas proteins and exactly what their PAM sites are. So different Cas proteins have different PAM sites. So it could be that by using a combination of different Cas proteins, you can expand out the ability to target different sequences around mutations. I don't know that you would ever truly be able to reach universal, is the only thing with that. There are other synthetic biology recognition kind of gene circuits that are more universal to SNPs, and we're working on some of those in our lab. So there was one that was published earlier this year called Inspector, and it's basically based on a ligation event that happens at a mutation location. And so the ligation will not happen if that specific mutation is there, and you can target it to any sequence around it. So it's not limited by any kind of PAM site or anything like that. And so there are more universal gene circuits that are capable of targeting targeting SNPs. Now, the trade-offs there are the Inspector circuit, I think, requires a mixture of about 4 different enzymes, and it's a more complicated design, right? And so your reagent costs then start going up too. The nice thing about the Cas reactions are their simplicity, right? But I think that there are other options in terms of being able to universally detect mutations as well.
Well, thank you so much for answering all those questions. Unfortunately, we're out of time. So as we wrap up today's PHO Microbiology Rounds, I want to sincerely thank you, Nikki, for presenting. I'd also like to thank everyone who joined us for today's webinar. You can expect to receive a brief and anonymous PHO Microbiology Round survey for today's session. Please, I do encourage you to complete it, as it really does help our programming. A reminder to everyone that TOPIC, the Ontario Public Health Convention, will be held on March 26th and April 3rd in 2024. Please check out topic.ca for details. And lastly, to access past PHO presentations and to view confirmed and upcoming sessions, please visit the PHO website, head to Education Events, and click on Presentations. With that, thank you everyone again, and I hope you all have a very wonderful day. Take care.